Hi, my name is Hassan Farouk, and today I'll be presenting a micro lecture on the hormones leptin and ghrelin. I will also be walking you guys through and explaining how these hormones regulate our food intake and affect our metabolism. So what are leptin and ghrelin? Leptin is an adipokine because it is secreted by our adipose tissues, and it is also a peptide hormone. It was the first adipokine that was discovered in 1994. This leptin, being an adipokine hormone, can act locally or systematically by carrying information or signals to organs, especially the brain or other tissues, to signal them about the adequacy of energy reserves that are stored in our adipose tissues, which are stored in the form of triacylglycerols. Leptin is 167 amino acids long, and it is located on the 7th chromosome. Leptin is predominantly produced by our white and brown adipocytes. It was first found in the obese or the OB gene of laboratory mice. Before I talk further about how this gene was found in the mice and how it it had its effects on these mice, before that I'm gonna touch base on the hunger hormone, which is the ghrelin hormone. It is a gastric peptide hormone that was discovered in 1999. It is relatively very small in size. As you can see, it is just 26 amino acids long. Ghrelin is predominantly secreted by our stomach cells, although it can be secreted by other cells such as liver cells and placenta. So as I mentioned in the previous slide, leptin was discovered in the OB gene of the mice. The researchers found that mice which had defective OB genes, the OB lowercase genotype, this resulted in elevated cortisol levels in the mice. They grew uncontrollably, as you can see in the picture in the bottom. They grew 3 times more than the normal mice. They were also unable to carry out the processes such as thermogenesis, so they were unable to keep themselves warm, and they were also unable to reproduce. So what the researchers did was they injected leptin into these mice that had these defective OB genes, and they saw some striking results. They saw that after injecting leptin, these mice ate less, they lost weight, and they also regained their locomotor activity as much as also processes such as thermogenesis were regained and increased. So these mice were back again to normal and were able to regulate their metabolism and keep themselves warm by thermogenesis. So when we eat food, the energy from the food is stored in the form of triacylglycerols in our adipose tissues. So as we eat more food, the adipose tissues will release more leptin, which will perform two functions. As you can see on the diagram on the right, the leptin is going to inhibit the synthesis of fat so that no more fats are being produced because adipose tissue is already full of these triacylglycerols. It is also going to stimulate the beta oxidation of fats. So leptin regulates the metabolism by increasing the rate of fat breakdown by inducing this beta oxidation of fatty acids. So leptin, when released in the blood by the adipocytes, it is going to first inhibit fat synthesis. So when you eat like a high caloric food, leptin is going to inhibit converting all that excess glucose into our fatty acids because it is already full of all these triacylglycerols. And it is also going to induce this beta oxidation, which is going to result in fat degradation. So these two processes are going to function together simultaneously and will decrease the amount of triacylglycerols that are present in our adipose tissues. So as we know that the more fat that is stored in our adipocytes, the bigger the cell gets, and this leads to obesity. So by decreasing the degree of this fat synthesis, less fat is going to be stored in our cells, and concurrently stimulating this beta oxidation of fatty acids is going to prevent obesity in our body. So leptin receptors are expressed in arcuate nucleus in our hypothalamus. So when adipocytes secretes this leptin, it is going to travel via blood, as you can see in the diagram on the right, and it is going to bind into the arcuate nucleus present in our hypothalamus. So our adipocytes are also innervated by beta-3 adrenergic receptors, which are activated by norepinephrine. So the arcuate nucleus, when this leptin is going to bind to the arcuate nucleus, it's going to signal the release of norepinephrine, which is going to activate this beta-3 adrenergic receptor, and a cascade of events will occur. So here I'm going to show you how this cascade of events is going to occur. So when the beta-3 adrenergic receptor is going to get activated, it is going to further catalyze, actually activate adenylyl cyclase, which catalyzes the conversion of ATP to cyclic AMP, the chem molecule. The chem molecule is further going to activate the protein kinase A, which can act and have a bunch of different reactions. The protein kinase A can go into the nucleus and facilitate the transcription and thus the translation of the UCP1 protein, which is the uncoupling protein. This is going to increase the expression of UCP1 gene, which is going to produce more UCP1 proteins, and these proteins are going to form channels in the inner mitochondrial membrane that are going to allow protons to re-enter the mitochondrial matrix without passing through the ATP synthase complex. This is going to allow a constant oxidation of fatty acids in brown adipocytes without requiring the need of ATP synthesis, which is going to produce a large amounts of energy in the form of heat. Protein kinase A can also go into the lipid droplets, and it can hydrolyze triacylglycerides or other molecules such as DAG and MAG into fatty acids, and these fatty acids are then going to enter into the mitochondrial matrix where beta oxidation is going to occur, and by beta oxidation, the energy released by this beta oxidation is going to be dissipated as heat. So leptin and ghrelin further stimulates the production of two types of hormones in our arcuate nucleus of the hypothalamus, which are going to regulate our food intake and metabolism. So leptin, as you can see on this diagram, leptin is going to activate the alpha melanocyte stimulating hormone, which is an anorexigenic, which means appetite suppressing. It's going to activate it, which is going to cause the second order neurons to send signals to our brain to stop eating and metabolize more. And this is going to lead towards the beta oxidation of our fats. On the other hand, you can see ghrelin hormone is going to activate the appetite stimulating hormone, which is the NPY, also known as the neuropeptide Y hormone, which is orexigenic. Which is orexigenic, which means it is appetite stimulating hormone. So this is going to signal the brain to eat more and metabolize less. Therefore, any signal which is going to activate the anorexigenic cells is going to inhibit the orexigenic cells, and vice versa, as you can see in the slide. Like leptin, when activates the alpha MSH, but at the same time, it inhibits NPY. So this is what I mean, that if the signal is going to activate anorexigenic cells, it is going to inhibit orexigenic cell. Leptin uses the signal transduction pathway, the JAK-STAT pathway, to regulate its gene expression. This pathway is also used by interferons and other growth factors. So, as you can see here, there are two leptin receptor monomers that are located extracellularly outside the plasma membrane. So when leptin binds to the receptor, which is also known as the LEPR, the receptor is going to dimerize. Both these monomers are going to get phosphorylated on their tyrosine residues by an enzyme called Janus kinase, the JAK enzyme. These phosphorylated tyrosine residues of the leptin monomers are going to act as a docking site for 3 proteins, which are the STAT proteins. The STAT proteins are called the signal transducer and activator of transcription proteins. The 3 docked STAT proteins are then going to be phosphorylated by the same JAK enzyme. This is going to result in their dimerization, and the STAT proteins are then going to move to the nucleus and bind to specific sequences on the DNA. And this is going to upregulate the expression of peptide hormones such as POMC, which produces the alpha MSH hormone. And as we studied in the previous slide, the leptin uses and acts on this alpha MSH hormone so that it can signal the brain and tell it to eat less and metabolize more. Leptin and ghrelin also affects AMPK, which is the adenosine monophosphate protein kinase. So let me just show you. As you can see in this slide, when leptin produced by the adipose tissues, it travels via the blood and it inhibits the AMPK present in the hypothalamus. Whereas when ghrelin, which is secreted by the stomach, it travels via the blood and it activates the AMPK in the hypothalamus. So the activation of the hypothalamic AMPK by ghrelin signals the brain to increase the feeding behavior and thus allows us to eat more food. AMPK also have different functions in different organs. So I don't want you to get too bogged down by all these details. Let me show you here. I just want you guys to look at how AMPK affects its target tissues. So as you can see, when ghrelin activates the AMPK, it acts on the brain and it stimulates the feeding behavior. But AMPK is also activated by leptin, and that is in the skeletal muscle so that it can stimulate fatty acid oxidation, and in liver cells so it can decrease the fatty acid synthesis, as we studied before. AMPK is also regulated by the enzyme acetyl-CoA carboxylase, which produces malonyl-CoA, which leads to fatty acid synthesis. And this is why leptin is postulated to inhibit this acetyl-CoA carboxylase, which would inhibit the production of malonyl-CoA, and thus it's going to inhibit fatty acid synthesis. And like we studied before, leptin inhibits fatty acid synthesis. So when leptin is going to inhibit this hypothalamic enzyme of acetyl-CoA carboxylase, it's going to inhibit the fatty acid synthesis, and this is going to inhibit our intake of food. Ghrelin receptors are located in hypothalamus as well as pituitary gland, also in other tissues such as heart muscle and adipose tissues. Ghrelin stimulates appetite as well as it stimulates the growth hormone in the pituitary gland. So the pathway of ghrelin hormone is not well studied. However, it was found that ghrelin acts on a G-coupled protein receptor, which further activates the second messenger molecule known as IP3, which helps mediating the action of ghrelin. As you can see on this graph on the right, the concentrations of ghrelin, they wax and wane throughout the day. So when you wake up in the morning, you're really hungry and it's time for breakfast, the ghrelin concentrations are really high. But as you eat the meal, as you eat your breakfast, the concentrations are gonna fall back down immediately. So this is how the concentration of ghrelin are gonna fluctuate throughout the day. High levels of ghrelin in blood leads to Prader-Willi syndrome. This is characterized as people who suffer from this are going to have an uncontrollable appetite and are going to be extremely obese. So this is the end of this micro lecture, and I hope you guys liked it. If you guys have any questions, please feel free to ask me. Thank you again.